I will do a quick run through of the TVB components at a maybe a slightly lower level than what Spasa did. That means that there will be a little bit of repetition, but we found that repetition legitimizes, repetition helps to understand things a little bit better, because the first time you see something, it might not be clear, and you can also stop me and ask me a question and say, that's not what Spasa said, for instance. Um, as Spasa said, we have usually done a lot of stuff inside the eBrains collab, but recently we noticed sometimes there are some hiccups with the eBrains collab. So today what I have done is to create there's a copy of these files in the eBrains collab, but there's also a copy available outside. And notably, this QR code, if you can scan it with your phone, it will give you a link, and that link will provide you with all of the materials. And it also gives you links to a copy of these files, which are in the, this is on the Google Drive. And if you don't have, or you can't get an account on the eBrains Collaboratory because your institution is not whitelisted, or you don't feel like it, but you do have access to Google Drive and Collaboratory, you can take these notebooks here and right click, and you do open with, and then you open the Google Collaboratory, and you'll be able to look at the notebooks. You might need to install some dependencies. But since today's Tuesday and we are leaving on Friday afternoon, you'll have time to ask for help. I'll be here around, and I'm here to help support you use TVB. So please ask me questions during the presentation or during the next few days. So you will be able to make this work on the Google Collaboratory, in the eBrains Collaboratory. And I will be running, during this session, I'll be running the notebooks on my laptop. So obviously I can help you run this stuff on your laptop. It's just that we found between Windows, Mac, Linux, there's always something to trip someone up. So I can come back to this QR code later on. At the end, I'll show it again. But now I think I can get started. This notebook that I'm showing here is in the format of slides. So I'm now going to go into the slides mode. So this is the second part of the TVB tutorial. I'm going to speak quickly about some of where we started with TVB, how to get up and running, components, what kind of data we work with, the resources on eBrains, and I'll mention the showcases, which are demos of TVB on eBrains. So again, please stop me, ask questions. These links here are available in the notebook, so you don't need to write them down really quickly. If you got the QR code, you have access to all of these links when you open the notebook. As Spasa mentioned, we have a mailing list, there's a website, there's the GitHub repository where you can report issues. You have a bug, you have a question, you can interact directly with us there. And there's also on eBrains a chat. If you want to get set up to run this, you can download from the virtualbrain.org. As Spasa mentioned earlier, you can open on eBrains. You can install locally rather quickly with Conda or the PIP tool. All it really takes is to install a few dependencies, TVB library notably. And once that's done, your notebook can start with just a few imports. Now, we've been at TVB collectively for over 20 years now. This started with Victor Gierza back in 2002 with a model of neural field model, whole brain model of MEG during a finger movement task. And the interesting takeaway from this, one of the interesting takeaways of this paper is that it produced interesting patterns of activity, but there was not sufficient symmetry breaking present in the neural field. And the authors said we need to have whole brain connectivity to make the activity more realistic. But whole brain connectivity, white matter tract connectivity, was not available at that time in 2002. It then later became easier to derive this from various data sources like anatomical tracer in the case of the COCOMAC database, and others started using diffusion imaging for human whole brain white matter connectivity. And those connectivity matrices, which I will show later, and you saw also with SPASA, are the way that we characterize the effect of the regions between the effect of each region on the others in the whole brain model. So there are a few studies in the before 2010, and that's where the scientific theory foundation of TVB was started, and then the development of a neuroinformatics platform based on that was funded, and the first workshop was done in 2014. And since then, there have been a handful of papers on different topics, from tumor to mouse to DBS stimulation. These are all links in here, again, in the notebook. So if you grab the notebook, you don't need to search Google for these author names. You can have the link directly to the paper, and I'll show the link again at the end. So let's take a tour through TVB. Again, this is a chance to see a second time what Spasso showed earlier, a little bit differently, ask questions if something doesn't make sense, okay? Neural mass mean field models are one of the primary tools used in TVB to abstract this complex mass of neurons into a tractable computational description of the activity. So we assume that there's an infinite number of inhibitory, excitatory neurons interacting in a specific way. We pool them all together and we average their activity into a few state variables. Some of the earliest examples of this were the Wilson, Cowan, and Jensen-Ritt models. The Kuromoto model of synchronization is a widely known model which can be used for neural modeling. More recently, we have an epileptor model of seizure dynamics, the Monbrio model of neural populations, and then since then we have more and more complex mean field models. This is the example of the Monbrio model, and again, as Spaza showed, this corresponds to differential equations which describe the evolution of the states in time. And the above model, those differential equations turn into a small piece of code like this in TVB. This is the way that we describe the neural mass models in Python code so that they can be used with TVB for simulation. All of this is fairly straightforward. It's a little more verbose than when you write the differential equations, but just to give you a quick read through here, we're creating a new Python class. We are making a list of the state variables. We need to say how many we have, and then we say which of those state variables is used to couple the one region to another region. In this case, the index of 0 tells us that it's the R variable, the rate variable, that is transmitted in our model from one region to another, not the membrane voltage. We need to specify reasonable bounds on those state variables. For instance, the rate shouldn't go below 0, and we expect that it's probably not above 2, and so on for the voltage. When we do forward modeling, by default, we're only going to use the rate variable, but this is something that you can change when you write your code. Next, we're going to define the set of parameters. In this case, I only defined parameter eta. In many neural mass models, there are lots of parameters. I think the Chromoto model is the only one where there's one parameter. The rest will have 5, 10 different parameters. And they can have default values. And then the last thing is that we need a computational implementation of the differential equations. And here, this is fairly straightforward. This is just these mathematical expressions written in a NumPy Python compatible format. So we have essentially metadata here. We have metadata on the model, which describes the model. We have an implementation of the differential equations, and that's all you need to tell TVB how to simulate a neural mass model, which again, the neural mass model is the description of the dynamics of a single unit inside the network. In TVB, there are 27 built-in models, and there are still several other models out there that haven't been implemented in TVB, but it's fairly simple to implement your own. And we have some built-in facilities in TVB for doing different things with models that so when we write our model, like on the previous slide, we can then very easily do a simulation with it by simply building a simulator and specifying our model like so. We run it, and then we can plot the resulting time series. Again, this is a notebook, which is in the folder that we're sharing, so you can download this and you can run this and try it out yourself. So where does the data come from? The data that is used to construct the neural mass model, the whole brain model, can come from different sources. So typically in TVB, it's important to understand that the brain is broken down into pieces by using an atlas. The atlas can be can involve very large parcels, like in the case of the atlases used by FreeSurfer by default. So the Desiccan-Killeni, the Destrieux, they have 76 regions, 164 regions. And the regions are relatively large. There are other atlases that break down the brain into hundreds of or thousands of regions. So when we want to work with a specific model, we need to also know what atlas we're choosing, because that will determine what connectome we can use. You choose an atlas, and then that determines what kind of connectivity, because the connections are between the regions in the atlas. So these data can come from different sources. They might be MRI data that you acquired yourself, and then you analyze with FreeSurfer and MRTrix. But you can also find data sets on the eBrains platform that involve different acquisition protocols in humans, but also different species. So, for instance, there's some mouse data. This is human data. There are also microscopic slice data that are available, but we won't work with those right now. The whole brain data are used then to build this whole brain model shown in the middle, where each red ball here corresponds to one of the regions, and then the connectivity between those regions is visualized with the green bars. So let's okay. So I said this again. There are some links here in the notebook that you can use to go find further resources to have more information. So one of the things one of the most important things so after we choose the neural mass model, the dynamics of the local unit in the neural network, the next most important thing to understand in a whole brain model is how they are coupled together. And this seems pretty obvious until you start looking at the different possibilities. So I created a specific diagram to show what this looks like, or at least what's happening in TVB. In TVB, this coupling occurs where we have all of the neural masses in the network have some coupling variable, which is then considered which I call pre here. These are all summed, and then we have a post summation expression that we can use, and then that enters into the neural mass on the arriving side. And this split between pre-summation computation and post-summation computation allows us to capture different effects. Sometimes there are presynaptic effects that you need to capture. Sometimes there are postsynaptic effects that need to be captured, like adaptation. So when we look at the different modeling papers in the literature, we can break down almost all of the cases in this way. So, for instance, in the case of the Kuromoto model, we are subtracting the difference between the sending node and the receiving node, and then we take the sign function of that, and then we do the summation, and then it's normalized. So we can create these pre and post summation coupling functions to capture the way the Kuromoto model is set up. But other models are set up differently, like in the Janssen-Ritt case. This would be a little bit different. Again, we can write all of this out in relatively simple code. We create a class. We have some parameters on the class, and we create two functions, pre and post, which implement the mathematical form of the coupling function that you want to implement. The importance of coupling shows up in almost every study on TVB, because once you've understood how the local dynamics, the parameters of the local dynamics, like the Montt-Brio model or the Janssen-Ritt model, once you understand how those parameters change the dynamics of that model from fixed point to oscillation to something else, the next thing that you're going to study with the model is the effect of the coupling parameter. So when we increase the coupling between regions, what happens to the overall brain dynamics? So here we're just showing a fairly stereotypical result where we vary the conduction velocity between the regions on the x-axis. And on the y-axis, we vary the coupling strength, and the color characterizes the standard deviation of the resulting simulated time series. And this is done for two data sets. So this is the kind of plot that we're going to compute when we're developing a new model, and we're not sure yet how to choose the coupling scaling. We're going to run a bunch of simulations, and we're going to look at how stable is the model with respect to these two parameters to try to understand how to set the coupling. This is another example, but in the interest of time, I'm going to skip it. So we just spoke about coupling. Now I'm going to talk about connectivity, which is very, it's close, but not exactly the same. The coupling is the mathematical form. Like in Kuromoto, you subtract the pre and post, you take the sign, you sum, and then you divide by n. The connectivity, in the case of connectivity, we're talking about the weight matrices, which tell you exactly how much does each connection contribute to the overall sum in those equations. Oh, sorry, I wanted to stay here for a second. So here is a nice illustration from one of the first papers on TVB, which illustrates that in TVB we are modeling the regions. The regions in the brain can potentially be modeled as a bunch of smaller units. So within the V1 cortex, for instance, we know that there are many cortical columns. And these cortical columns can have local interactions, which are visualized here. That means that this vertex here can interact with the vertices nearby, but it doesn't interact locally with vertices that are farther away. Now we also, that's something we call local connectivity in TVB. And then the counterpart, global connectivity, is going to be the non-local connectivity that goes through the cortical white matter fibers, or cortical, subcortical, and so on. So in TVB, we can have both kinds of connectivity, the local, distance dependent, and then the long range uh, connectivity, which is derived from anatomical tracer or diffusion imaging. Let's skip that. When we install TVB, we have a few default data sets for the connectivity, and those can be initialized as in this example here. Again, you grab the notebooks, you can run through the examples, and then you don't need to remember exactly what's written here, but you'll have the example that you can run yourself. When we initialize the connectivity for the white matter, we also specify a conduction velocity. And as I showed you before, the conduction velocity has an impact on the stability of the model. It's a parameter that needs to be taken into account as well. Again, we have a few different sources of data. So this is an example of what the connectivity from diffusion imaging looks like, but there are other resources on eBrains and outside of eBrains for getting connectivity data that we can discuss after the talk if you have specific questions about that. Again, there's some links that can help you get started, and I'll let you read those if you're interested. So once we've described the differential equations and the coupling, we have integration in time. And what that means is that TVB doesn't simulate the, sorry, let me step back once. The differential equations describe a continuous time process. In TVB, on a computer, we can't do continuous time. We're doing time stepping through, we are stepping through time to do the simulation. And one of the things that has an impact on the simulation results is the integration scheme, which is used. It's a small detail, but it's worth paying attention to. For instance, the time step that you use needs to be adapted to, for instance, the time scales which are present in the dynamics. So the most commonly used scheme for our models is the Hoyne scheme. The Hoyne scheme that we have implemented in TVB, it takes into account the regular differential equations, but also the noise and the time delays. There are other schemes that are better if you don't have time delays. There are other schemes which are better if you don't have noise. But the Hoyne scheme works quite well for the generic cases of noise and time delays, and it's simple to implement. So this is the one we recommend you choose when you run the notebooks and you're doing your simulations. You can, of course, implement, as with the other components, you can implement your own scheme relatively easily. If you've done any kind of differential equation stuff before, you're probably familiar with the Euler scheme, which consists mainly of evaluating the defun, taking the existing state, and then adding the dt-weighted derivative. And so this is what the Euler scheme looks like in TVB. If you can use the Hoyne scheme, you don't want to use the Euler scheme. So this is just to show you what it looks like. Again, there are a few more resources here. One thing which is worth keeping in mind is there's a few steps on choosing DT. You start with a small value and then you incrementally raise it until you see that the results are qualitatively changing it, until you see that the results are changing qualitatively. Sorry. Then we would also advise if you have, a major figure for your publication, you might want to try to reproduce it with a lower DT just to make sure that when you come back to it later on, it's still, it's still valid. Okay, forward solutions are what we call the way that we take the neural signals generated by TVB and we generate neuroimaging signals that then are comparable with what we record in a human. So this is going to be fMRI, MEG, and also intracranial EEG. These are examples of initializing these different monitors. I don't want to spend too much time on this because it's in the demos and we mentioned it earlier. So I'm going to skip through this a little bit. You have more links here, and all of the demos will show you how to use these monitors. Yeah, okay. Stimulation. So, right. So in TVB, a lot of the first demos in TVB were oriented around spontaneous activity. But we also have several use cases that make use of stimulation. And so in TVB, we can set a spatial stimulation pattern. This is visualized on the left, and a stimulation time course. Those are specified separately through a spatial weighting pattern and then the temporal pattern. And then we pass the stimulus to the simulator, and then it's used during the simulation. So this is an example, and there are a few more resources here. Once we've built up this simulation, and that's one, and it just does one, that generates one set of time series, then we want to understand how does this depend on the parameters of interest. And for that, we need to do parameter sweeps. But we also need to choose a metric that could be the stability as indexed by standard deviation or other metrics like metastability. But it could also be something about that characterizes how well does the simulation match some empirical data. So in many studies, the correlation between empirical functional connectivity and empirical simulated functional connectivity is used as a metric. So then this bump, when it goes up, it means that the those two FC matrices match a little bit better as a function of the parameters. So that's a fairly easy technique to do. We can discuss the details a little bit later if you have questions. But okay, about half and half here. We have, since about 10 years, been working more and more on doing proper inference with TVB models. And that means that instead of just trying to guess the parameters that make the data, the simulated data look like the empirical data, we're going to use Bayesian techniques to invert the model and identify what distribution over the parameters is consistent with the empirical data. And there's a small point, but an important point there, that optimization and Bayesian inference don't do the same thing. Optimization of the parameters will give you the peak of the distribution over the parameters, but that peak is not necessarily representative of the full distribution. And in high dimensional space, actually the peak of the distribution doesn't lie where you expect it would. So that's why when we have high dimensional parameter space, such as estimating a parameter per region in the brain, we need to use proper Bayesian inference and not just simple optimization to find the parameters of interest. So what this means is that we're going to do some simulations. The inference algorithm is going to look at the data feature. So in this case, we have some time series that's being simulated, and it's going to then identify distributions shown on the right here over the parameters, which allow the model to be consistent with the data. So here, this is a little bit. it's a little, sorry, I can't zoom in here. We're going to go into a concrete example in just a second. But in blue here, we have the empirical data, and in red, it's the time series generated by the model. Some short practical advice. If you only have 1 to 3 parameters, you can still use grid search or parameter sweeps. If we're up to 30 to 100 parameters, then you can use techniques like simulation-based inference, which we are going to see in just 1 minute. If you have many, many parameters, then one needs to look at more challenging inference techniques, such as variational inference, which is what the DCM toolbox uses, or MCMC. And so a lot of these concepts are discussed in Maysam Hashemi's paper. Maysam is sitting over there, and you can ask him many questions over the next few days, or both of us. Just to put you on the spot. Thanks. So right before we go into the notebook, I'm going to introduce roughly the overall workflow. The idea is that we're doing in simulation-based inference, we are doing lots of simulations, and we are training a neural network to generate a posterior distribution over the parameters, which such that this distribution is consistent with the data. And as we increase the number of simulations, we get more and more accurate results. And we've shown that this gives us good correspondence in the application for clinical epilepsy. So let me switch over to an example of that, which is small enough or tractable enough that you can run it in the eBrains Colab. You can also run it on the Google Colab, but it also runs quickly on a small laptop like mine. This is is the text, can you tell me, someone in the back can say, should I zoom in a little bit? Would it make it easier to read what's on the screen? Okay, let's do that. So I'm gonna do full screen, and then is that gonna be good? All right. So this is a inference demo for the virtual epileptic patient, which is an application of Bayesian inference with TVB we have been developing for almost 10 years now. And in this workflow, we're going to show you how to use the method of simulation-based inference to do inference for the parameters of this model. We have I won't show you the equations for the mathematical models, since we just went through a lot of that material. We have a specific nonlinear neural mass model. We have the connectome of the patient. And now we're going to step through the process. Again, all of this is available on the link that I showed you earlier, and I'll show the QR code again so that you can download all of that. To do this, we're going to need a few utilities, some of the typical stuff, but we have a specific package that we have been developing called VBI, Virtual Brain Inference. And this is a package which is dedicated to SBI-based inference for virtual brain models. So this is a package which is going to be interesting if you want to apply this to your data sets. So we're going to import all of this. And to ensure that you can reproduce the results, the figures that I'm showing you exactly, we're setting the random number seed. That's not necessary if you're just working at home by yourself. We have some, in this case, the data set is fairly simple. We just have a connectivity weights matrix, which we're going to load. And I kind of want to know how many, 84. If we have 84 nodes, then we're working with the Desikan Kilini atlas. So when we develop a model, and it's a model of a pathology, we need to decide how are we, yes, please. So in this case, this would be it's a good question. Let's just
sorry. I was asking what's the range of scale of these weights, the connection weights, if they are probably that's affecting the accumulation there. So
I'm trying to.
I mean, you can just comment on it. That's like I'm.
I kind of wanted. I was kind of curious myself what the range of the weights was.
Thank you very much.
So the 5th to 95th percentile is like this. It looks like this might be fairly logarithmically distributed, which is fairly typical for diffusion-weighted imaging. If you're not familiar with diffusion-weighted imaging, what happens is it measures the diffusion of water. That image is then being traced, and we count the number of traces we find between the regions. And those are typically very logarithmically distributed, which means that some connections have 1,000 times higher weight than other connections, which is why, as Pazda mentioned earlier, we might use some kind of normalization because it doesn't make sense functionally that 2 or 3 connections within the brain would dominate completely the dynamics of the brain. So we might take a log scaling so that a higher percentage of the connections can participate in deciding how the dynamics unfolds in the model. In this case, in the demo is not written with much thought to this particular question. So I'll have to gloss over it a little bit. As for interpreting those weights themselves, I would recommend looking at the literature. I'm not an expert on the diffusion imaging topic myself. We tend to, so again, the answer is that we tend to interpret the connectivity weights themselves in terms of how they impact the dynamics of the whole brain model. Okay. Yeah. Yes, please.
I have also a question. Biologically, I'm not an expert. How many states weight can have a synapse have? I mean, there's probably a certain resolution biologically that you cannot have on 3 decimal accuracy of synapse. So, what is known? What is the accuracy of these weights biologically?
So the highest resolution that I'm aware of, based on diffusion imaging, results in a connectome that has a few 100,000 nodes for the whole brain. And that threshold was chosen by, in terms of reliability of scan, rescan. So you do several scans of the same brain. You process all of those scans with the same pipeline, and then you break down the parcellation, the atlas. You break it down into smaller and smaller pieces, and then you ask at what point, from one scan of the brain to the next one, do you not get the same results? And at some point, the connections between 2 parts of the brain, they stop having the same statistical distribution, and that's what they consider to be the barrier, the noise barrier. And so below that, we don't have good, and so that corresponds to about 0.5 millimeter square on the cortex. And so that's still quite a lot of synapses, yeah. Are there any other questions? Okay. So when we start building a Bayesian model, it is very important to choose how the model is parametrized. What that means is that the inference technique, whether it's optimization or Bayesian inference, has to work in a particular parameter space. And how that parameter space is chosen will determine whether the overall method is going to work or not. So in this case, what we are, the way in the VEP, the virtual epileptic patient, in the VEP models, we are parameterizing the pathology of the epilepsy as a single parameter of the neural mass model. And this parameter is interpreted as the degree to which the model will initiate a seizure without any noise input, so just spontaneously initiating a seizure. That's in terms of the nonlinear dynamics, there's a critical value that you determine through a bifurcation analysis. But if we zoom out of that and just look at this in terms of the brain modeling, we consider one parameter per region in the brain, and we say that there are, through the bifurcation analysis, we know that there are specific values that we can interpret as healthy, what we call a propagation zone, which means the seizure can propagate there, but it won't start there. And then the so-called epileptic zone, the seizure will start there if we set this value in that region. So those are specific parameter values that we use to model the seizure. Then we set up spatial mask. That means that we choose specific regions which will be epileptic, specific regions which will be the propagation zone, and then we assume that all of the other regions are healthy. Now, why do I say that we are setting parameters here? In this demo, we did not include intracranial time series from a real patient. So what we are doing here is a simulation. We're going to generate a synthetic seizure, a data set, and then we're going to run inference on that synthetic data set. And that means that we have the ground truth for the parameters and the time series, and that allows us to check for ourselves that our inference technique is working the way we expect. Because if we look directly at empirical SEG, intracranial SEG, which presents a seizure, we can run inference, but we don't know if the result is right or not. And so to confirm to ourselves as a sanity check that it's working, we're going to do a simulation, and we're going to check that we recover the parameter values that were used for the synthetic data that we are trying to run inference on. So that means that we're going to have a vector of values called eta true, which are the ground truth values for this parameter. Next, we're going to initialize the dynamics, the simulator. We have a simulator for the VEP model, and that's going to run. And then we're going to get what I was saying before, a synthetic seizure, which is plotted here. And this is what we're going to run inference on to try to identify the parameters that were used to generate this. Are there any questions at this point? That's just an excuse for me to drink some water. Sorry. Okay. So just as a quick reminder, model inversion proceeds, it's not a reminder since I didn't say it before, sorry. Model inversion here is going to proceed with the following steps, and I apologize for reading the text, but that's I'll try to be brief. We're choosing relevant data features because if you choose the wrong features, then there won't be any relation between the parameters and the simulations. We need to choose a prior distribution over those parameters. We're going to run a lot of simulations. This part is really key in this workflow. We run a lot of simulations where we sample from the prior distribution of the parameters, and we compute the features on the generated time series. And this allows us to sample the effective relation between parameters and data feature values. Because we assume in our workflow, in any kind of inference workflow, we assume that there's a robust statistical relation between the parameters of the model and the features of interest. Otherwise, the features are wrong, the parameters are wrong, the model is wrong. So this is a strong assumption, but I want to highlight it because it means that in your workflow, when you're building a model and you're not yet sure what are the relevant features, what are the relevant parameters, this is what you're looking for. You want to find features, parameters, which allow you to build a robust statistical relation between the two. Once we're pretty sure that we have the right parameters and the right data features, it's relatively simple to then train a regressor. In this case, it's going to be a Bayesian regressor, which maps data features to an approximate posterior density. That means that once you compute your data features, you can then obtain easily a posterior distribution, which tells you what you should expect those parameter values to be. And then optionally, you can apply that regressor to other data to do new inferences. So now I'm just going to show you, we're going to go quickly through the steps of what I just explained, which are facilitated again by this toolbox VBI. There are feature extraction elements in there. Then we're going to specify the prior distribution. And then we're going to, there's a little bit of utility code, which you would probably be able to just copy paste into your workflow if you chose to, which allows to run the simulations on all the cores on your computer. So we run 1,000 simulations, and then we have, as we said before, we have 1,000 instances of the samples from our prior parameter distribution, and we have 1,000 instances of those vectors of the features. Then we can train the Bayesian regressor, we save the result, and then we can check whether we got the result that we used for the simulation at the start. And that's shown here. So in this workflow, we try to identify 3 parameters. Oops. Let's see if I can just, okay. We try to identify 3 parameters. G is the coupling scaling between the nodes. Eta 1 and eta 2 are 2 of those parameters for the degree of pathology. And along the diagonal of this set of plots, we are plotting the distribution over the parameters themselves. And the red bar here indicates the true value that was used for the simulation. And the black line plots the distribution of the values found through Bayesian inference. And off diagonal, we plot the joint densities between these different parameters. And so in this case, we can be fairly confident that the inference worked well. We can only do this because we're doing inference on simulated data, but this helps us go through all of the concepts and check our model is working. So that's where we stop with this specific example. If you are interested in understanding more about how do you validate it, how do you validate these inferences, how do you check that they worked on real data, there's a literature on that. I've put 2 links here at the bottom of the notebook and you can go to, and you can get a lot of material there to help you understand how would you validate a result once you obtain it on some real data. And that's it for this demo. We have 6 minutes left, so we could have a few questions if you like. Yes, Mike. You are not allowed to ask questions without a mic.
Okay, thanks a lot. So, but are we allowed to ask generic questions as well?
You can ask any question you like.
Okay, thank you. So, from the language technology perspective, so in both of the cases, from the large language models perspective and in here, the ultimate goal is to create the model of the brain. So, but I see that the architectures that we are using in language technologies and architectures and equations you are using in here are so much different, but we are still thinking about the same brain. So, what would be your ideas? Maybe one day all it would collide into some model or thing, or because we are using different granularities, and as well with LLMs, we expect to be it not just the average human, but like the humanity brain or humanity knowledge. It is not even possible. So,
Okay. So that's an interesting question. One that we have thought a little bit about informally. But I think two comments to make there. One is that the goal in TVB, so the kinds of models that we build with TVB are distinguishable from the kinds of models built in machine learning in general, in that we would call our models parametric models, whereas in machine learning, and especially in LLMs, they are essentially non-parametric models. They have parameters, but you don't try to interpret what their values mean. In our models, we absolutely want to interpret what the values mean. And if we cannot, then we say this is not a good model. Does that make sense? So that's one comment. A second one is that because of that bias and how we construct the model, we do not need to have enormous amounts of data to do something useful. Now, when you look at building an LLM, why does it work? It works because the essential ingredient is the self-attention mechanism in LLM, which is a nice, clever trick, but it's still a non-parametric modeling trick to create something that scales well. So while I do think that there are several parallels between the brain and the LLMs, for instance, the self-attention trick is something that allowed transformers to continue scaling when other architectures would not scale. And by scale, we mean with compute and data size. The cortex was the first structure evolutionarily. The cortex was the first structure that continued to scale. All of the other structures, a bigger amygdala doesn't give you a functional advantage. A bigger thalamus doesn't give you a functional advantage. A bigger cortex explains most of mammalian evolution. Everything was about having more cortex. That's why the cortex is folded. So I think that there are some parallels like that which are worth exploring. But at this point, when you look at the models which are being built, like what we're doing and what is in most of the LLMs, I think that they are driven by very different use cases, and they're not yet convergent. The last point that I would make is that there are some attentional mechanisms recently published that have tried to derive a recurrent neural network approach. And there we start to see how we could do something where we take that mechanism and we could fit it into a TVB-like architecture, and then do something where we have the TVB style dynamics and the anatomy and so on, but we would also have enough parameters and enough expressivity in the model to capture cognitive tasks. But we're really at the point where we're drawing the diagram and trying to connect the dots.
Okay, thanks a lot.
So just here's the QR code. If you didn't get it earlier, you can go there, and from there you have links to Google Colab, eBrains Colab, and you can also just download this stuff and find me afterwards, and I'll help you get it running on your computer. All good? Thank you very much.